The human brain is a box of mysteries that we might never fully understand, and its ability to make complex decisions in milliseconds is baffling. However, progress in research and development in the sciences has helped us gain a clearer understanding of how the brain makes decisions. Our lives are defined by the little decisions we make every day. We are forced to choose between numerous possible options and situations, and act in the way that is most appropriate to achieve the chosen reality. From choosing what to have for breakfast to selecting a life partner, our chances of having a good life depends heavily on our ability to make good decisions. It is estimated that, on average, we make thousands of conscious decisions on a daily basis. Some of these decisions are made very quickly, as fast as milliseconds. Some, however, take time and a great deal of thought. A lot of our decisions are made based on past experiences and learned information. Anticipated rewards often influence our decisions. We tend to make decisions based on a set of actions that we believe will lead to a desired outcome or at least prevent undesirable events and situations. The brain is a vast repository of information, and its neurons hold data that for the most part cannot be accessed or even understood. Our inability to decipher that information that is held in the neurons has restricted our understanding of how the brain works and why it stops working. This understanding will help us learn how the brain's neural circuits process and store information. The human brain and mice brains have similar decision-making structures, and the functions of the neural circuits are quite similar as well. Like humans, mice have the ability to make sophisticated decisions and can be used for difficult decision-making tasks. Mice use visual cues and their knowledge of their environment to create a cognitive representation of the world around them. They then determine the best decisions to make based on the likelihood of reward or punishment. Decisions that trigger a stronger activation of the brain's reward system are reinforced so that we are more likely to keep making that decision so that the rewards keep coming. Decisions that lead to a negative outcome are less likely to be repeated. Recent imaging studies in humans have also shown that the activity of dopamine neurons, which are functional cells in our nervous system that release the neurotransmitter dopamine, in the orbitofrontal and posterior cingulate cortex of our brains is heavily based on the expected rewards that have been associated with a decision. Decision-making in the human brain is a cognitive process. The process of making decisions involves a number of stages. Each stage can be considered a cognitive criterion. The brain uses its knowledge of the criteria to predict the result of an action. The knowledge is located in the hippocampus, and this information is processed in the prefrontal cortex, which performs the function of approximating goals. The prefrontal cortex and the hippocampus are necessary for decision-making activities. The decision-making process has 4 main steps. The first step is the production of initial stimuli by sensory inputs. This triggers a set of hippocampal neurons as part of the neural system. The second step involves another set of stimuli which enter the hippocampus and start a stimulus-driven neural response. The third step involves the transferring of the initial information to the prefrontal cortex, which then determines if additional information will be needed and retrieves complementary information from the hippocampus. The final step occurs when the prefrontal cortex decides the best action to take. The communication between the prefrontal cortex and the hippocampus is achieved with neural connectivity. The neural wiring creates closed-loop neural circuits that generate decisions. The frontal lobe is believed to be responsible for thinking, planning, and making decisions. Damage to this part of the brain has been known to negatively affect a person's ability to think, plan, and make decisions. Figuring out the precise set of actions that occur before a decision is made is key to curing a number of brain diseases and increasing human potential. Many of our best examples of brain activity during the decision-making process are obtained from extensive research by distinguished neuroscientists. A great example of this is the work of Antonio Damasio, which has shown that patients with damage in their prefrontal cortex exhibit impaired emotional responses, which often leads to them making bad decisions in risky situations. Silicon probes have also proven useful in gaining new insights on this topic. They have the incredible ability to record the brain's activity, which makes it much easier to study it. These silicon probes are very thin, thinner than human hair even. Silicon probes can have up to 1,000 recording sites, which enable us to record information from more neurons than was possible in the past. Prior to the development of silicon probes, we could only record a single neuron at a time. Now we can record more neurons than we thought possible. We can now study how different parts of the brain interact and how information is shared between them. Though we've made tremendous progress, we're still just able to observe a small amount of the active neurons involved in decision-making. Further development of the technology available currently will help us gain a clearer understanding of the causes of disease such as Alzheimer's, autism, and schizophrenia, and increase our chances of finding suitable cures.